Welcome everyone to the second virtual biology meetup. Today, as you can see from the cover, we will talk about agents. This is a very old agent from Star Wars, which is appropriate because we have the 4th of May today. The topics will span from virtual cells, virtual biology, even more mechanistic simulations to agents and how we can make them understand biology and talk biology. And it's my pleasure to introduce the two presenters who we have with me today. One is Theo from Achira AI, who was before that head of AI at CZI and also VP of AI in Citro. And Daniel, who is leading digital biology at NVIDIA, and before that was doing single cell research at Celerity and Open Problems. So it will be a very fruitful discussion. I really hope that we will tackle some interesting topics, which will be also very practical, because I'm almost certain that everybody on this call is using some kind of agent. So first we will start with the presentations. Theo will kick off, and then Daniel will follow. Q&A will be in the end after the two presentations, but you can already submit questions into the chat. And yeah, let's kick off. Theo, the floor is yours.
Yeah. Hello, everyone, and thank you very much for that invitation. It is always a pleasure to be with this group of people, in particular whenever I can share a stage with Daniel. It's a fun day. So I hope to be able to do this justice and everyone will have some fun. So I'm going to talk a little bit about world models at the example of biology and how I believe they set us up for working on scientific intelligence, which no doubt everyone will connect to agents as well. And because we don't work on science to just be machine learning people who have fun with models, you can do that in many domains. I want to start with why should we be working on these things? And there's certain bottlenecks in the world right now. And these bottlenecks are very real. For instance, here, there's the bottlenecks that we really have in biomedicine. This is very actionable and vivid in the case of target discovery. We are not doing a great job as humanity really exploring the richness of the human genome to really understand how much of it is actually targetable, how much of it is intervenable, and how much of it we can understand mechanistically. And, a lot of us have been talking about various aspects of how data generation, AI, and various things could come together to tackle this. But this isn't just to do science, but this is very much how can we accelerate the progress towards finding intervenable and actionable means to cure disease. And this is in the targets. However, the same bottlenecks also exist right when you have a target that you believe in and you want to make drugs. If you think about this, drug-like chemical space is vast. It's gigantic. It's so vast, it's not enumerable in experimental space. So search in experimental space for drug-like molecules just does not scale. We have ample ability to make drug-like molecules, but which ones should we make? Which ones should we try? And there's a lot of these ideas around high-fidelity molecular simulations. It's a wonderful path. We believe in it greatly at Achira, but in the past, they've been too expensive and fragile and haven't served as a universal screening engine, which we're about to change. But the bottleneck here is that small molecule discovery fundamentally is bottlenecked. Not bottlenecked by the number of molecules we can imagine, but by the ones we can really simulate and make in the lab. Now, how does this constitute a more abstract problem fundamentally in life science and physical sciences today? The discovery loop, the scientific loop, just does not scale. We're doing amazingly at them. We have wonderful data generation modalities. We have very smart analyses now. We have all of these things, but our approach towards tackling these things, these domains, hasn't quite reached the industrial scale yet that we have in, say, tech and in the digital domains. And that looks like we do 90% of work in the lab. I don't know if it's 90%. There's a huge amount of work that happens in the lab, although I will say in the last months, I don't know if these numbers are still up to date. A lot is happening that makes me very happy. But discovering new treatments for diseases fundamentally still is a slow process. We can't run experiments on humans preclinically. We have to wait until we find out what happens in the clinic, and human disease biology is still vast and unenumerable. So we do need to transition into this paradigm where instead of hoping that we do discovery, we do design. And the idea of how to actually move towards a world where inverting biology and life sciences from discovery to design has to go through building world models and has to go through a process where we build models of the processes we care about such that we can basically perform hypothesis search in silico and then utilize the lab for validation, for scaling of hypotheses, and for fine-tuning, and actually for teaching models new tricks. And what we believe here is that it is essential to create virtual biology simulators spanning multiple scales and enable what-if scenarios that are very hard to scale in physical labs. Not every question you can formulate and think of is easy to directly answer with an experiment in a virtual lab, in a physical lab, but in a virtual lab, surely they should be. Now, I want to also take a bit of a historical lens here. Biology and life sciences have captured computer scientists since the beginning. I would say that Claude has been working on biology since before he was working on information theory. Maybe we can close that loop and get back to it. I think that's happening. And John von Neumann also was extremely interested in self-reproducing automata because he realized that information is preserved in the real world in interesting ways that are tremendously robust. And how would we map this in a computational way to life? And I think this is exactly what we're getting back to. It just took us, 100 years to build the technology stacks that we need. So it's time for a model-based paradigm in cell biology and the life sciences that really embraces this idea of hypothesis, experiment, and update through models. And, we had one Perspective article reasoning about this a while ago. But let's just quickly talk about what is a scientific world model. We've had predictive models in computational biology forever. The idea of a predictive model is you have some quantity that you care about Y that you can just predict with a model F. But in the scientific paradigm, you really care about things that represent also the latent state of objects. And then you want to be able to intervene on that latent state in order to find out how can you transition from that state to a new state. And this allows you basically to do the core thing that we care about, which is virtual experiments. You want to be able to simulate what will happen if I apply a particular change to an object. And this doesn't necessarily need to be one model. It can be a system. There can be multiple things. But fundamentally, the idea really is that you want to capture dynamics. You don't just want to capture a static state. You want to be able to intervene on that state and capture dynamics and link them back to observations, such that you could basically say, I did something to my latent state, to my cell, to my human, to my molecule, and now I get these new observations. When you can do this, you can actually move to this world where you don't just predict, but you can simulate. This was also at the heart of this idea of virtual cells that we kicked off a couple of years ago. Right when I started at CCI, we gathered a lot of our friends, Daniel included here, into a room for a few days and tortured them and wouldn't let them out until we had the core ideas together for what this paradigm of virtual cells would look like in the AI age. We were lucky enough to get that organized and published in Cell. But the core idea here is a lot of people are pursuing this. This became relatively loud, which is a fun thing to be able to enable to happen. But the fundamental idea really is that all modalities and scales matter, and that we want to build these very general, very powerful models that will capture a lot of that knowledge and information in cells. And how do we start? What are key modalities? I will leave this for another day that's only focused on virtual cells, but the idea really is we need to start from what we have in genomics and transcriptomics and work towards generating data sets and building models for richer things at the interactome that really will have more mechanistic ways or richer ways to model how things really interact and bring all of these things together to characterize cell states and characterize how these systems behave when they're kicked. And everything has a place in here in terms of the different layers of information between sequencing and imaging and proteomics and many other modalities that people are coming up with. But again, this is for another day. The core philosophical, but also very practical idea after I introduced the world model is that whatever models we build, we want them to be able to act as virtual instruments. So we have to be able to ask these what-if questions and answer them and act on them. And this is going to be a core message here, because if you can simulate in silico, you can search in silico, and then you're still reliant on the lab to perform search. And that is a key thing that we want to be able to change. So I will talk about instantiating a few of these world models in biology at the example of virtual cells, anchored on a few of these works that we built with my previous team at CZI, and frankly also before that at In Sitro, but I can show a lot about that work. And I'm going to focus a little bit on introducing these three models that really live in two categories: this transcriptome and single cell latent diffusion model, SCLDM, which are models of transcriptomics. And then this variant former, which is a very different model that also predicts transcriptomics, but it is based on DNA variation. And if you wish, the first category of models is transcriptome as modeling variation in gene expression across evolution, all species, all cell types, all cells. And it's trying to characterize what is the richness of individual cells across bodies, development, and really evolution. SCLDM is really targeted at being a very, very powerful virtual experimental tool that is able to generate experiments, that is able to generate outcomes when we simulate conditionally, where we say, do this to a cell. Under this condition, what would happen? And in the future, these may be connected. But variant former is a problem that really maps to a different problem because it stops looking at all of evolution and so on and really maps to population scale. So it tries to really understand variability on a population scale, mapping from genome to phenotypes, which is basically the idea: how does individual level variation captured by allelic variation, so the mutations that make us up, also lead to the richness we observe, which is a very different idea than what is typically studied in this nascent field of virtual cells, but really is at the heart of understanding variability as well. And it's closer to how people have been reasoning in medicine and genetics. So transcriptome briefly is this big model where we try to model all the data in the cell atlases that we could find that had sufficient quality. This is a model across evolution, across development, across different species and cell atlases, in order to say, if we trained on all these gene expression patterns that we can find, can we build a foundation model of the variability we observe in these cells? And this is an object that we ended up throwing 12 different species in there, a lot of different cells across 1.5 billion years of evolution, really. And it models genes and the expression values in a joint generative process, so that you can really think about a constructive process, building cells, and build a model that represents cell state in such a way that you can then use in a variety of tasks. Cells here really represented as bags of transcripts. They're not represented as structured objects that have an order in these transcripts, because transcripts really are bags, but we're not modeling where in a cell these transcripts are expressed and so on. So there's no spatial notion here yet. It's like a histogram of our expression, which is still, if you wish, a very early vision of how cells will behave, but it is this joint cellular vision where we model gene expression for a cell. And one analogy, to not get into too many details, that I want to make is that this is very much 2018 in AI. Because in AI in 2018, we started building transformer models right after transformers came out across many different languages. People started training transformer models across different languages and realized that as they do this, they can start to do unsupervised machine translation. And this is a very similar paradigm here in the analog to biology. If you start training across species, which much like language live on this tree of differentiation of where the variation comes from, you can start learning to connect that information in interesting ways and building analogies. So we're starting to trail only 8 years behind, at least in this paper, behind AI. And I'm sure that later Daniel will show us that maybe we're a little bit further ahead than I think. And I'm not going to go into details for transcriptome, but you should look at the paper. We also have a fun version of it coming out very soon and an accepted version. Then in SCLDM, we went both one step back and one step forward. SCLDM does not have the ambition to model all of evolution and all of the cells anywhere, but it is for a focused gene expression world. Really, can we build the most powerful generator that does virtual experiments that will capture this conditional nature? And here you can really go in and say, given that you have a certain cell state, which very much could also come out of transcriptome if you wanted to, how can I go in and decode this to a new or to an expressed cell state? It's trained as an autoencoder, but you could really use this as a conditional generator in interesting ways. This is a paper coming up in ICML 2026. And the hallmark of this is really that it creates extremely high fidelity generations under different conditions. So this is in some way, if you think about these two works, are in some ways your virtual experiments for gene expression. So you can take these models and really think about, I want to explore how does gene expression across cells, across evolution look like? And I want to start asking questions. How can I start generating really targeted profiles if I wanted to explore new ones? And in the literature, it is exploding with many different works exploring that interface. I'm of course going to mention my team's work, but this is a very rich field. There's a lot of work studying these interfaces, this idea of perturbation biology, this idea of conditional generation, and so on. And it's, I think, a wonderful space where we start having more and more of these world model structured objects that can start reasoning about perturbation biology under structure. But then variant form is a very different category, which studies variation where, if you wish, your perturbation or the changes you induce, the actions you induce are mutations, which typically aren't studied in the cell biology field. And here we're thinking about, there's been a lot of structure sequence models like EVO2 and so on that have been reasoning about, can we capture the heterogeneity and variability of genomes? Even alpha genome does that, right? On reference genomes, how can we model these things? But none of these have really started reasoning about what do individual alleles do in real human populations. So variant form is the first model that is really trying to capture the full variability of human population variations. Yes, I see a hand. Should I be stopping to answer or?
No, you should go ahead.
Okay, good. So here we are asking the impact of allelic variability on cellular phenotypes. We can learn across all human tissues. Let me just give you the quick kind of visual view of this. You can really think of this work as something that goes from personalized genomes to gene expression across population. So you can go for a sequence that has all these individual mutations, not just in the exome, but across the entire genome, everything, regulatory gene aspects of the genome everywhere, across every tissue. So we can take a genome and then really per tissue make predictions for how this really looks. And this goes all the way, it zooms from an individual genome to the expression in an entire human. And this is how you can model variability across different humans. This is not a clinical tool, mind you, but this is an ambitious foundation model that tries to match this entire degree of variation here. And ultimately linked to diseases. And of course, the way we built this is we took inspiration from the mechanism of gene expression and then built a hierarchical transformer architecture that tries to go into these things. I will ask you to read the paper to see about how we had to do this, because the main challenge that transformer models have had is how can you put so much of the genome into a transformer model that you can really capture this allelic variability to model these differences, including the regulatory genome. And then you can do fun things with it. You can use this to try to model and predict personalized gene expression predictions. So what would happen in a particular what happens in a particular person across tissues in terms of gene expression with a high likelihood if you do this. And this model is the most predictive one we could find until this point on these tasks. But you can also do this for this virtual experiment where you can say, I can do in silico mutagenesis. I can flip an allele and say, if I changed something, what would happen? If I changed 3 alleles, what would happen? And in a world where very much we would like to explore the effects and understand these, you can do this very nicely and start building these models that try to predict what is the causal outcome of flipping an allele, having learned those outcomes from populations, model them, and then maybe you might go back to the lab to cells to try to engineer these types of things and really try to see in the lab what effects you can see in appropriate cell models. Now, this is great. And these are, in some sense, examples, instantiations of models that start approximating the idea of world models in biology. Some of them in the idea of gene expression variability and perturbation in terms of extrinsic variation. variant former in case of modeling genetic variability. And if you could engineer that, what would be the outcomes? But now, how does this map to this idea of agents and learning from them? And here I want to introduce a third thing we did as a proof of concept, but there's examples of this that are starting to abound, which is where you can start combining this idea of these world models and biological data with reasoning models. And the work we did here is RBio 1, where the idea was that we want to teach agents to reason and talk about cells. And the core thing we did in this work is that we used world models of biology as environments to teach agents. So here they are not used as just data to supervise them, but you can literally simulate data, just like we explained with world models, and teach your agents based on simulated data. This is the paradigm here. And so you combine the world models that we've established and built with reasoning models here. And then RBio is a bridge between those, and we introduced a couple of concepts here with reinforcement learning with experimental model feedback, because you're not just generating outcome, but really supervision in terms of rewards for your models, and also reinforcement learning from prior knowledge. If you put all of these things together, you start getting a workflow that looks like this. So if you go from world models to an environment for agents, this looks like you have a world model, you have a simulated environment, which is, for example, your world models of biology, your virtual cell models, in the future, your Achira models as well, if you care about molecules. And your agents here will start then making hypotheses, running virtual experiments, updating themselves in order to learn. And ultimately, you will have the real lab, and the loops here are wrong. The loops will go, the world model gets better from the lab model, and your agent gets better from the model by training on the world model. And there's a lot of different details about how RBio does this, but fundamentally, as I said, RBio abuses the world models as training ground. It uses them as simulators so it can expose itself to see basically as much biology data as it wants to see by asking world models questions instead of relying on real data. We have a few paradigms in here and an example in perturbation biology. And lo and behold, the results say it actually works. It starts reasoning purely by training against simulations. It learns to reason about cells really well at the example of perturbation tasks. It improves if you do chain of thought type reasoning. It improves on models just trained on experimental data. It beats models like Summer, which was another more retrieval-based model from Genentech that tried to do these types of tasks. And it even learns zero-shot skills. For instance, on data sets it has never seen, never trained on, it learns to do disease prediction in a stronger way. At the example of, for instance, an Alzheimer's data set or myeloid data set. So it gets zero-shot disease prediction skills if you just ask it questions there. In the interest of time, I will also start getting to my last few slides. What might the future look like? We will be combining not just the models that we tested this early version with, but all of our different models. So this looks like virtual biology at that point, where you really take agents that learn from many of these biological world models across all data and all scales, and really integrate that knowledge together into these big objects that start understanding biology jointly in multiple layers. Then I also want to quickly explain Achira. Achira is the idea that you're not just going to rely on these, if you wish, biology scale abstract models that really try to build transitions, but trained very close to the data that they can generate. Achira is going to be building a real mechanistic layer for virtual biology, and I think of this as virtual chemistry. So if you can learn the physics of how things interact on a molecular level, you're really building a very powerful simulator. And Achira is going to be working on transforming drug discovery into drug design by really modeling these interactions on the molecular level, on the full molecular level, and the complexity that they have. We think of this as building microscopic world models, where you take the richness of quantum chemistry, the richness of molecular dynamics and statistical mechanics, and you cook this into very rich world models that will start capturing the energetics of this world such that you can really at high fidelity simulate data sets. And Achira also has some interesting, we have job opportunities, we have interesting things to share, and just check it out. And in the interest of time, I think I am at time, and want to really thank my team at CZI, that is now BioHub, and my wonderful team at Achira for a lot of work we have been doing and we are doing. And I look forward to sharing more on the panel.
Thank you, Theo. This was a wonderful overview of the field, and I think you were very nicely leading up to Daniel's presentation. As I mentioned, discussion will be after the two presentations, so please submit questions. We already got one, which we will try to address in the discussion, but don't be shy and submit more and more questions. So Daniel, you are coming up next.
Perfect. All right, you all can see this okay? Awesome. Hey everyone, so my name is Daniel Burkhardt. One quick correction, I do not lead digital biology at NVIDIA. That's my boss, Anthony Costa, but I'm a developer relations manager. So I work a lot with companies building biofoundation models and especially doing agentic AI in the life sciences. Today I want to open this presentation by asking a question: what if doing science were as easy as vibe coding? Before we get into that, I want to just maybe give people a little bit of an understanding about what developer relations is, because it isn't something that I knew before I got into NVIDIA. I work sort of as the interface between the product teams that build libraries, SDKs, open models, and the ecosystem. So NVIDIA does not sell these tools, but we want to understand what's useful, how do we know where the field is going? And so my job is to work with companies like BioHub or like Achira to understand, hey, or Turbine. Speaking of which, like, what are you guys working on? What's hard? How do we build the right thing for you? So to start this presentation, I want to share a little personal story. In the fall of last year, I started using a tool called Granola. I am in a ton of meetings every day. Like in a given week, I might meet with 20 different customers and need to understand, like, what are they working on? What are the follow-ups? Who do I need to introduce them to? And having a tool like Granola was really a game changer for me, because this meant that I didn't have to be scribbling down notes in a notepad. I could just have an AI assistant that was listening to all my meetings and would write summaries based off of it. And in about December of last year, I got this lovely notification popping up on my computer. It turns out that this was not an approved third-party LLM tool at NVIDIA. And I was told I had to basically stop using it, which made me deeply sad. Around the same time, this is like, winter break, right around New Year's of last year, I saw these two quotes, one from Andrej Karpathy, one from Yana Dogan, which both basically said, like, hey, AI coding tools are here and they are fundamentally changing the way we work. I thought when Andrej Karpathy is saying, I feel so behind, like, that's a signal that's worth listening to. And so over the holiday, I decided, okay, can I go and build a tool to replace Granola for me? So I did what any good person does and opened up Cloud Code and said, hey, create a clone of the Granola app for me that runs locally on my Mac and does transcription. I want all native macOS UI components and a really nice user experience. I wanted this to be a test because I actually know no Swift. I used to do Python programming, but it turns out on macOS, if you want to get like a really nice user interface, you basically need to write in Swift. I thought, what better test than, like, can I build an app in a language that I don't even understand? So I'm not going to lie, this is not like a weekend project. It turns out there's a lot of parts about dealing with microphones and speakers and trying to do echo cancellation and handling multiple devices that took like probably about 6 weeks. And I feel like 1 billion tokens might actually be an underestimate for this. But at the end of it, I built a tool, and it's called Muesli. And it's a local meeting transcription tool for macOS. There's now a bunch of these that are out there, like Open Oats, but at the time, no one I knew of had built anything like this. There was like Whisper Kit or something, but I want something that was slick and I want to do it myself. This was like a really great, I think like a really great experience for me. I built this tool, basically able to just like create, meeting transcriptions. I knew that this was something that was actually useful when I got a message from my boss. This is the actual head of digital biology at NVIDIA, who actually started using this tool. And when he said that actually it saved him in an important meeting, I was like, okay, like I, who knows nothing of this programming language, was able to build a app that my boss uses and finds useful. I was like, this is something that's really interesting. You can go and use this today. I'm going to be honest, like I've stopped maintaining this a few weeks ago because we now do get access to some of those tools again. And this is, I definitely think, one of the things I would rather buy than build, especially like the point I got tearing my hair out was trying to deal with echo cancellation with Bluetooth speakers and a USB-C webcam on my home setup was just like not working. And I was like, there are, acoustic engineers who solve these problems. But if you want, you can use this or Open Notes or any of these other, tools. This really got me thinking though, like, okay, this is very different. I had in grad school at my previous job, I was writing a lot of software. I was even doing the tab completion stuff in Cursor. But the experience that I had going from just, hey, this is an idea about something that might be interesting and useful to building something that actually other people would start to go and use made me start to think, okay, the world of writing software has fundamentally changed. What does that mean for science? Because as a computational biologist, like I, in my last job, did a very similar thing to what Theo was doing, working on a research team, trying to help people build models. All of my day was writing code, right? The thing I was doing were tokens of software. But there's a little bit of a difference between engineering and software. I want to I guess maybe this will sound somewhat familiar to folks here, but want to just kind of tell a story of well, what would actually have made a difference for me. So this is a paper that I worked on before I joined NVIDIA. It was published in Science last summer, doing single cell analysis for understanding whether or not you could identify a small molecule that would induce a complex phenotype, like induce directed differentiation, basically. The work of generating this figure 1 may look familiar to some people here. This is actually what happened. It looks really nice and neat, but basically I spent, maybe 1 or 2 weeks. So each of the column here are weeks. This is not days. I spent a couple of weeks QCing a data set, running some clustering, creating a first draft of the figure. The following week was able to meet one of the biology leads. I don't really know anything about hematopoiesis or CD34 stem cells. So basically they're like, hey, this is kind of interesting, but your clusters are totally off. And also there's some cells here that may look like a separate cluster, but they're actually dying. You need to remove them. So I spent another few weeks doing this process with them. And they're like, well, can we look at the site-seq markers, or can I look at the chromatin accessibility? This was a lot of effort just into getting what is the label of the cells. Then one of the people in the team said, you know what, maybe these aren't really clusters. Maybe these should be trajectories. Can we actually try to, do this totally other kind of analysis? Great. All right. Turns out, since I was a graduate student, there's 4 new packages to do trajectory analysis. Wanted to pick up one of the new ones. It's academic research software. It really sucks. I spent a lot of time debugging dependencies. Then I show them the trajectories. It turns out they actually like the clustering better. So now we go back, but the cell annotations are wrong. Basically this is literally 12 weeks, 3 months of my life was just basically taking a single cell data set and getting it to a point where an expert biologist thought that this was actually, usable for them. And this, I think is a really good example. That is not useful time, in my opinion. I don't meaningfully better understand hematopoiesis. Maybe a day's worth of learning, I better understand it. I also don't think that the cluster definitions themselves fundamentally changed things. A lot of this was helping biologists get comfortable with the data and understand what it was that we'd actually measured and trust the experiment themselves. So I think there's a lot of time here that just was total process waste. Some is valuable, to be clear. I'm not saying you shouldn't collaborate or you shouldn't talk to biologists or biologists shouldn't talk to computational people, but there's definitely some opportunity here, I think, to improve things, both for the people who are writing code and for the people who are doing experiments at the bench. I think that if doing science were as easy as vibe coding, you can imagine now the biologists that you're collaborating with are actually enabled to explore data on their own. So rather than me being the gatekeeper for this new trajectory analysis algorithm they want, or the cell annotation data, they can just go and ask an agent to go and analyze this data for them using tools that they've never been trained on before. One of the things that Jensen has said repeatedly that has always stuck with me is AI is one of the only technologies that actually teaches you how to use it. And so if you have a coding agent and you say, hey, I don't really know what this does, or I don't know how to use this tool, you can just ask an agent. It'll tell you. This is a fundamentally different kind of technology. And this also enables them to get sort of input much faster to design their own experiments. I think this also is incredibly valuable for the people who are sitting, working on code every day. I don't think that they're going to go away. In fact, I think we're going to need more of them. And I think that the code that they're writing is going to get more reliable and reproducible. I guarantee you, if those trajectory analysis packages were written by Claude code, they'd be much more likely to work again in the future because they have like unit tests by default. I think that, generally find this to be true. They can also scale out experiments. They can start to tackle harder problems outside of their domain using tools or theories that they're not necessarily so familiar with. I also think that one of the things that's interesting or exciting in a future in which doing science is as easy as vibe coding is that you can start to get to this world where you can actually have fully autonomous labs. Like you can have labs that are taking some, maybe like to start simple, analyses totally out of the human loop and just, there's something that happens in the, 3 a.m. in the morning. Maybe an experiment has an anomalous result and you need a decision. Do I want to start the next experiment at 3 a.m. or do I want to wait until like 4 days later once people are able to actually like get time to analyze it? I think that stuff's going to really change fundamentally the way we do science, though I'm not going to talk so much. Happy in the panel later to ask more about this, like totally autonomous self-driving labs, but for now, I'm not going to talk too much about it. So great. This sounds wonderful. I wish I lived in that future 10 years ago, but we're not yet quite there today. But I think that we're going to. I definitely think that the path, though, is not yet quite clear. It's certainly not just taking the exact same kind of frameworks that we have around agentic software engineering and trying to get them to work, perfectly for science out of the box. And the reason is because writing software and doing engineering and doing scientific discovery are just like not even close to the same process. Like, yes, this observation I started with, hey, I used to do computational biology. I wrote a lot of code. Software engineers also write a lot of code. Doesn't mean that the thing that they're doing, the actual process is the same, right? So in engineering, for software, or for anything else, the goal is to solve a problem. If you build a bridge and the bridge falls down, you did not solve the problem. You failed in engineering. But in science, the goal is to discover new things. The goal is to identify like something useful. So if you have built a bridge and the bridge falls down, but now you better understand the theory of bridges, that still can be a success in science, even in engineering that like maybe just doesn't matter at all. And it's also like science is like fundamentally a unbounded process, right? I don't think there's ever going to be a point in which we truly understand everything that there is to know about the universe. And so until you have that point, there's still a frontier in science. Whereas like in engineering, like there's a concept of over engineering, right? There's not a question of over sciencing, right? There's a definite end point for software. Okay, so what are the actual kinds of problems that we need to work on, and how are the ways that I think that AI is going to start to influence them? One of the fundamental differences about doing science, I think, Theo mentioned this a little bit, is that in order to discover something new, you need to actually know that it's new. You need to be able to go through and understand stuff that has been done in the past. I did not believe this chart the first time that I saw it, but there are over 3 million peer-reviewed publications every year. Like, that is insane. How on earth are you supposed to go through 3 million new articles to understand is something that you're interested in discovering actually part of our understanding or not? And so one of the things that I've worked on along with a partner of ours, Edison Scientific, they used to be previously known as Future House, is helping build agents for doing literature analysis and document understanding. I definitely did not believe when I started working on agentic AI that the thing that would be hard about doing science is drawing bounding boxes on text and figures, but it turns out actually it is. On the left, you can see here a benchmark that we did comparing a few different kinds of agents, including a Frontier Labs deep research agent on a series of benchmarks that Edison has put together in basically a package called LabBench 2. And on the right, you can see an example of one of the outputs of a model that we've developed at NVIDIA called Nemotron Parse, which takes as input an image of a document and then draws these bounding boxes. Why this is important, if you use heuristic rule-based parsers and you don't know where to put this purple box around the image, the way that LLMs work today, you have to encode text and images separately. And so you need to know where in the document is the image. A lot of times what we were seeing is that maybe you would get a bounding box that would just focus on panel A1. And then if the agent wants to understand something that's in C2, or if there's a bounding box that cuts off in the middle, there's just no way for an LLM to reason over that part of the image. It's actually like a huge part of the improvements that we're getting here between, you can see like without, these are rule-based parsers versus our Nemotron parse implementation. This 15% increase is almost entirely due to just being able to draw boxes around PDFs better. Right? This is a fundamentally important problem for scientific discovery, not so important for engineering. Another, I think, interesting difference about science versus engineering is that in science, abstractions are to some extent the focus of the work. I think that Theo started to talk a little bit about this around the idea of like trying to understand the latent space, the latent state of a system. I think a lot of the work you see in like, you know, the figure 6 or 7 of a cell paper where you want to say, well, this is the data generating mechanism, even if we don't observe the whole thing directly, we're just taking these snapshots. Like that's kind of the work that we're trying to do. And you could see here about 30 years of evolution of understanding a tumor suppressor gene P53, right? There's like the first study in the 1990s of this has like a couple of interacting patterns and one regulatory loop. And now we're seeing like dozens of pathways, dozens of complexes that this has, you know, that this protein interacts with. And like to some extent, like this is the knowledge that we have around this. Like this is the progress that science has done, is our ability to represent this kind of abstraction. Because when you have this kind of abstraction, then you can start to actually reason beyond what you've observed by guessing sort of like what would happen if you knock out, you know, this pathway, for example. Taken to its extreme, actually, this is some of my favorite virtual cell work, which is really these like mechanistic virtual cells. Where it basically doesn't scale beyond bacteria or artificial, like synthetically minimal, like yeast cells. But this is a fully specified mechanistic dynamical model of E. coli. And there's a story that's like not really easy to see from here, but I'll tell you, that was discovered through this model, which was this interesting observation that they had where by simulating multiple generations, okay, let's say one step back. For E. coli, you almost never measure individual cells, you measure populations, but then you fit these parameters at the individual cell level. And what they saw is that at the individual cell level, although there are some genes that are lethal if you delete them at the population level, they're only transcribed less than once per cell cycle, which is this interesting question of how do you get, it's called subgenerational transcription. How does a cell survive without transcription of genes that are essential to the life of the cell? So they thought, well, maybe it's the RNA. You just transcribe a bunch of RNA and the RNA gets stabilized and then it gets diffused. And they looked through the simulation and they saw, nope, actually, like the RNA gets degraded relatively quickly. So they go further down the chain. Is it the proteins? Maybe you get a protein pool and then that sort of like gets diffused and they maintain these proteins around. Nope. Proteins also actually tend to get mostly degraded within a single cell cycle. And so they say, look at the very end of the pathway, which are the downstream metabolites of these essential genes. And turns out actually this new theory was developed around how, you know, cells sort of survive with subgenerational transcription of essential genes by preserving metabolite pools. So they create a whole bunch of the byproduct of some of these essential pathways. That byproduct would just go sort of floating around in a cytosol through sequential rounds of cell division. And this isn't something that had been experimentally described before and now is like generally understood to be part of the biology of E. coli. And this is only possible because we had sort of this level of diagram about how we think the cell works. We could then go and fit, you know, differential equations to all the parameters and then run these simulations over time. Like those abstractions are genuinely valuable for science. I also, let's see, I'm not exactly sure how this video will work. I want to tell just another quick story about Edison Scientific. This is a demonstration of an AI scientist platform. I want to really get you sort of communicate the value of being able to scale this discovery. So we're seeing here like a real user query about Alzheimer's disease. The agent starts by doing literature search. It has this hypothesis here. It's given an objective by this orchestrator agent. And then it goes and does a whole bunch of literature search. And you can actually go, I can share a link to this. You can actually see this run. It goes and looks through, basically like 45 million 75 million documents in a database that this agent has access to. And it's basically trying to say, do I find literature that either supports or refutes this hypothesis? Let's see. I think, how much more time do I have, Daniel?
Let's say like 5 to 10 minutes.
Okay, perfect. So I'll just finish through this then. Okay, so it comes up, it like does this literature search, it writes a little report, it says, yes, this is supported. Then it goes and does data analysis. So this particular task is associated with a particular single cell data set. Tell me, is the expression of this gene correlated with aging in this particular cell population? And it says, yes, it is significantly correlated. It goes through and actually generates a Jupyter notebook that has figures as output that you can see. These are like sometimes very, very long analyses. So this is like all an artifact that's generated by an agent that's running fully autonomously. So the way that this system works is you enter that initial prompt, you hit run, and then you come back like 24 hours later to a fully completed set of analyses. So you can go and see the reasoning chain of the agent. But this is just like 3 tasks. When you go and run this system, you get hundreds of tasks run fully autonomously. These are hundreds of hypotheses that are, gone and vetted either through literature search or analysis of data sets. And then the output is this report that you can see, which in my mind is like kind of, what you would expect a graduate student to provide their supervisor at their 6-month dissertation committee meeting. And that's how, basically how Edison came to this, headline number of, we have a system that can do 6 months of research in a day. Excuse me. They actually collaborated with a number of labs, did these kinds of studies, showing that about 80% of them are human reproducible. And the average sort of span it would have taken a standard lab member to complete the work would have been about 4 to 6 months, which I think is totally reasonable, seeing the amount of stuff that's happening here. Now, this is, I think, really cool because for the first time, you're seeing scientific discovery scaling with electricity rather than with labor. So a human plus a whole bunch of tokens and agents in a data center can go and do this, basically to the extent at which they're able to generate questions that they want discovery to happen over. While you're spending this 24-hour research job, you can spin up, 5 or 6 other, totally parallelizable processes to go and investigate anything that you could possibly imagine, get deeply cited, deeply analyzed research outputs. Okay, so I talked a little bit about the vision, the future, what are some of the things that are happening in the space today. I want to also talk a little bit about what NVIDIA is doing in this space, what we're really excited about. When I start to think about the components of AI as applied to science, this is my personal, sort of set of buckets. There's some other ones other people will use, but I sort of think of this reasoning layer. I think people always talk about a reasoning layer. What does that actually mean? This is the LLM. This is the context management. This is the improvement of the cognitive layer that takes as input text or images or featurization of molecules, what have you. And then basically decides what work to go and do next. And I think that in my mind, there's 4 major buckets of work. There's search or remember, or I don't know. There's a few different ways you can think about this. There's predictions, which are the application of ML models. There's simulations, which are the application of physics-based models. And then doing. So basically the affecting action in the real world. NVIDIA is working across this entire stack. We have open models like Nemotron that you can use as sort of an open weights alternative to Qwen or Kimi. We have inference servers, Triton, TRT-LLM, Dynamo. These are things you can use to actually deploy those LLMs on whatever hardware you have, and actually frameworks to go and train and build AI models. When it comes to searching, this is basically trying to retrieve knowledge that exists. We've got tools for doing graph-based agentic RAG. We've got enterprise knowledge retrieval frameworks. There are tools I mentioned before, like Nemotron Parse, that are really designed for document intelligence. There's a whole bunch of tools we've developed in a framework called BioNemo, which is our digital biology platform that has tools for things like protein structure prediction, molecular AI, some like single cell analysis, like data loader, stuff like this. There's also a bunch of work we do in simulation. So this is a little bit different because I think simulation is much more physics grounded. So there's a lot of work in quantum chemistry, like FEP simulations, climate simulations, also physical AI. So if you want to understand how robots are going to interact with, say, like a cell culture bag, there's a whole bunch of tools we have in Omniverse and Isaac. And then finally, you have to go and do stuff. And so I think that doing stuff can be either the like data analysis, like writing code, and then also robotics, right? So these are, I think, all fundamental tools that NVIDIA is working to try to build for. And these all fit into like, again, this is how I think about an AI scientist system. I've heard of a lot of people sort of describe these differently. There was a AI software engineer report that Anthropic put out, I think back in like February, that inspired some of this. Here in an AI scientist, like you have a human scientist that basically talks back and forth traditionally to an orchestrator agent. So this is where, in that example I showed earlier, you write a query, it goes into a search box, the agent goes and does a bunch of things. Within each of these agents, there's a bunch of fundamental primitives that all agents share. So there's the LLM, the intelligence, there's some system for context or memory that's more of like a database. There's a series of tools that enable the agent to basically operate in the world of bits. And then there's a harness that manages the behavior of all of that. I think what there's still a lot of question around is whether or not we will see a ton of variation in this specialist layer. The example that I showed you before with Edison Scientific did have specialists. There's a literature agent, an analysis agent, there's a molecules agent I didn't show you. But I think that there's also some groups that are thinking, well, do I really need that? Or do I just want like some sort of complicated memory system, plus just like the ability to write code and execute bash commands, I think is a big open question. But one of the other really challenging parts I think is still very much unsolved is, okay, like you can have this agent, you can have these tools, but to do all of those different kinds of things I showed you, from search to simulation, prediction, data science, you also need access to compute in some way. And these different kinds of agents are going to connect to compute differently. I think this is a very unexplored area today in agentic AI, is like, how do you make sure that specialists can leverage either serverless inference on modal, or can use your submit jobs to your HPC without spamming it? These kinds of things I think are still emerging, but are really important if you want agents to be able to do things that have productive value. And finally, like I maybe want to just take a step back and really emphasize that there's every layer of this stack is really important for developing effective AI systems and having economic value. And NVIDIA really tries to provide solutions throughout every layer of the stack. So you maybe are familiar with GPUs or networking that we provide, like InfiniBand, that enables you to connect many different nodes in a single rack, but actually not even know that there are separate instances. So you can just so that they're separate instances, but that they're not on different racks. So you get sort of transparent connection to many, many GPUs. There's also actually, I didn't know this, but NVIDIA acquired the company that writes Slurm earlier, or at the end of last year. So that's like another sort of open source tool that we really work to maintain because we think it's critical for AI. And this goes all the way up through libraries, SDKs, models, all the way to like supporting services, either like AWS Bedrock, which is where a lot of these LLMs are served, or really working to support the application layer. Because for a lot of people, we really think that if you want to have like widespread value from AI, a lot of that's going to happen through applications and products. Even now today, like I said, I could go and build my own meeting transcription tool, but I'd rather pay for it. I could go and build my own voice typing tool, but I'd rather pay for it because I actually just get a much better experience at the end of the day. And I don't want to have to spend, my weekends and evenings working on that. I want to go work on other things. And finally, I want to just emphasize something that I think NVIDIA has been really proud of, but maybe is not quite loud enough at. Well, I think maybe we've been doing a little better this year. But NVIDIA really fundamentally believes not just in open source, but also open models. This is, I think, this is definitely out of date now. This is from the end of last year. But just in terms of language models released open weights on Hugging Face, NVIDIA is the leader in the industry. And it's something that we are fully committed to long term. So please go and try any of the Nemotron models, try any of our BioFoundation models. It's something we really care about and we want to get feedback on. That is it. So thank you so much for the opportunity. I'll stop sharing here and excited for questions for the last half hour.
Thank you, Daniel. Let's start with your presentation, Theo, and I would first address the biological word model definition and what do we actually mean on them and what is the right resolution. Because when I see how people are communicating about word models, many are thinking about word models as basically a more global description of how cells work, and they can address like multiple angles from transcriptomic prediction to genomics, while your definition was much more narrow, that the word model is predicting one given task well, and then you can line these up and connect them through an agent, and then you can get to a more complete understanding. So my question to you, which kind of also leads up to your new work, Achira, but what's the right resolution you think to build these word models, and for what do we have enough data? And sorry, just one final thought on that, because we had a question on the scaling laws in biology. So what blocks us to create a word model which can address multiple questions? How do you see that?
Yeah, thank you for the question. I will unpack this. I think there's 2 or 3 questions in there, and remind me if I miss any of them as I try to go through this. So first of all, the vision we put forth for a virtual cell isn't one of 50 different models, but it's one of one unified model ultimately. However, we clearly don't have the right data for this. We don't have the amounts of data, but not even the right types of measurements to build this immediately. So I think that's a long-term vision. It's a long-term vision because we both need new types of measurements to begin with for the full cellular dynamics that we would want to have, but also at new scales, at new amounts of variability and so on. So if you then start thinking about and I had a slide, I understand I went through a lot of material quickly about where do you start? What can you do today? What should you do today? We are going to focus on the things that are scalable and where we can generate a lot of data in this cell paradigm. And I will switch to Achira in a second. In the cell paradigm, it is really important to think about where do we have good amounts of data sets, where can we generate them, and where are we unlocking more and more. So with my previous team, we worked heavily towards setting up new data generation. And, BioHub, for instance, recently also announced that they're investing a lot more resources into data generation and various things. And I think in the public space, this is wonderful. And this is exactly the way to go. We never have enough data already. We need to keep generating more and more and more to build these types of ambitious models. But also, in the commercial space, a lot of entities do generate data sets at industrial scale now of wonderful quality and depth and diversity and so on. And we're going to keep doing that. And not all modalities are equally scalable today, but things like sequencing and so on, single cell sequencing are becoming very, very scalable, and we start having a bounty of amounts of data. I think this is why we see so many models on that front, because the data sets are starting to be visible, at least. We're starting to be able to characterize. Now, your question, but what is the right resolution? The right resolution doesn't exist. There is no right or wrong. There's only what questions are you trying to answer? And is it appropriate to try to build, whatever you want to call a world model at that type of layer of abstraction? Because here's the interesting thing. Biology is very compositional. It starts from what we worry about in Achira, which is the fundamental physics of how atoms interact and compose to molecules, and molecules interact and so on, which is really on the level of physics. But then on top of that, nature has come up with biology based on a few different rules. It's one version of biology of the many possible ones, but this is the one we have on Earth. And within there's a whole space of things that compose, much like programming composes, right? And from very low level, byte level instructions, you can get to high level programming and to data prompting. And all of these things can characterize similar programs at different levels of abstraction. And I think depending on the questions you're asking, certain layers of abstraction may be perfectly sufficient to generate data and build models there to answer them, so long as your mechanism can be isolated to that layer of abstraction, if that makes sense. If you're trying to answer questions that depend on fine-grained causality that requires fine-grained knowledge, and you build macroscopic models, you won't be able to answer them. So I think that's the wrong level of abstraction. But if, for instance, you're trying to answer questions that do operate at some abstracted level, models at that layer will be helpful. So the point I'm trying to make is I believe people should not wait for the next 20 years until we have all the data for everything and everything is solved in order to start doing things. We have a bounty of data sets. We have a bounty of better and better models we're building. If I look in the last year or 2 years alone of the progress in building models of various types of cell modalities, it's staggering. We're really getting better at this. And lots of companies and public entities are generating data sets. And I think that's inspiring. And we should pursue that. And as we get more and more layers of data available, it'll get better. Now, generalization is a very big topic and a complex topic. And I think this is also fundamentally where this idea of virtual chemistry and virtual biology differ. Virtual biology, you really need to probe science and probe reality, measure cells, measure living systems in order to get these things. Your generalization will always be close to your training data because this is how it behaves until you've tiled the space enough. In chemistry, we have other advantages because it's physics-based. So there is a mathematical generalization layer there that we could get to that is more general purpose and more generalizable than trying to enumerate things. This is why at Achira, for instance, we're so fascinating to turn extrapolation problems into interpolation problems once we've mapped the physics correctly. And another thing there, because in your typical drug discovery campaign, you will make in the order of a few hundreds to a few thousand molecules, and the amount of public available data for really, binding affinity, for real chemistry events, touching biology, modeling proteins, interacting with proteins is very limited. You will never have the amounts of data sets you get out of single cell sequencing. The cost, we're orders of magnitude off from being able to just generate enough chemistry data to do this. So what you need to do there is you fundamentally need to build world models that will be data efficient, which is why we built these physics-based world models that really try to also be able to utilize synthetic data and simulated data and bootstrap themselves to generalization without requiring endless amounts of measurement data. So I think what I'm trying to convey is at each layer and for each type of question you're answering, your approach towards building a world model has to be fit for purpose. You cannot just say, I will take a big autoregressive LLM and train on everything and everything will work perfectly fine if you don't have the appropriate amounts of data. And you don't need to, or a diffusion model or whatever you have. So I think if you build the right category of model at the right layer, you can make a lot of progress, and this is not a pipe dream. And we're only getting better at all of these things. So I hope I answered a few of your questions, but let me know if anything is missing.
Yeah, you did a brilliant job, although I asked multiple questions in one question. And just to follow up on that, so what we see many times is that, rightly, as you just said, you can build fit-for-purpose models, like we call them virtual assays for that particular data, doing one particular job. But if you want to move towards a unified world model, which you mentioned in the beginning, like a long-term goal, then what we miss today is data which is connecting these different fit-for-purpose models to each other. Do you have any hint on what data is missing? Because it's not just the scale.
Yeah. One of the greatest challenges in measuring biology is that it is typically destructive. That means if you want to measure one layer of biology, you will never get another layer out of the same cell. That cell is gone. You have to measure that layer somewhere else. There are modern modalities where you get multiple layers. That's awesome. We love it. It's still early. So a big problem for these multimodal, rich, and in particular temporal models is how do you get these matching layers of biology? And I think that is something that we're starting to get to with imaging. Imaging is a very rich modality where you might be able to get extremely rich and multimodal layers of data sets, in particular in the future. But also, there is no need to wait until we have those matching perfect data sets, because you can integrate multiple different layers in different ways into single models. You can build multimodal models that are pre-trained in each layer in certain ways and combine them. There's things like late fusion you can attempt from small amounts of overlapping data. but also in the agentic way, I try to show that there's a possibility to bring together models that operate at different layers via some prior that unifies concepts. For instance, an LLM that understands concepts can bring these things together and try to interact with them this way. You can bring these tools together via using tools that try to understand how different layers connect. So I'm actually optimistic that this isn't something we have to give up, but we can't be naive and just say, we can train a model on everything at the same time, and there isn't much overlapping data there. So again, it's a bit of a non-answer in terms of do this one thing, because it depends on what are you trying to achieve. But I'm not pessimistic about it because we've made progress, and I see that there's a lot more progress being made. And if anything, what I see happening in measurement biology tells me we will get a lot of interesting bridge data sets, as I think of them, that can connect depth in individual modalities with bridging modalities together. And yeah.
Totally agreed. And with that, we can also bring in Daniel to the conversation, because you mentioned LLMs or agents, like connecting these different models to each other. When we are experimenting with like unified model for predicting dependency and transcriptomics, and also like single models. And what we see that there can be inconsistency between the models if you are predicting with one model, let's say the dependency, and with the other model, the transcriptomics. And that's something what you can better manage potentially with a unified model. If you have an LLM which is orchestrating all of these, then how do you look out for these inconsistencies, and how do you actually like meaningfully connect up these multiple models with multiple readouts? So the biologists are not just getting, a crap of signal which is very inconclusive, but to get and drive some conclusive hypothesis.
Was that for me or for Theo?
Yes, yeah. Sorry, I tried to prompt you to join us. Yeah.
So, yeah, it's an interesting question. I mean, I might like take a little bit of a step back. I think that I definitely get at, or I've thought a lot about it and asked a lot about like what is the interface between LLMs and biofoundation models. And I think like I kind of see them still at the interface they're basically still seen as an external tool. So I don't know that there's necessarily that's anything different from querying a biofoundation model to make a prediction versus like doing a database lookup. I think it's just like you understand that there's a slightly different confidence associated with it. Like, I don't know, Theo, do you agree with that? Like, I actually want to ask you a little bit about your thought here.
That is the canonical way, and I'm totally in agreement with that, but we tried something else with RBio. So RBio does not do a lookup. RBio uses the biofoundation models to simulate outcomes that are used as rewards to do reinforcement learning on the LLM. And then the LLM changes its weights depending on that. So you don't use them as supervision directly, you use them as a reward source, which is a bit different. And I think you can do both. And using them as tools is wonderful.
That's the training paradigm, though, right? Like in the discovery mode, right? So there's always different ways to think about this, right? So you have to but once the model is fully trained, like you want to stop doing, like probably you want to stop doing RL at some point, because like not all memory or not all knowledge of science has to be parametric. There's like a whole bunch of problems with having like all of your understanding of biology be embedded in the parameters of the neural network. So at some point, like, okay, now we're getting a little bit towards that like far future. Like I've got an AI science factory, or I'm like building a fully autonomous lab. I need to be like just doing at some point, like you need to do physical experiments. I think like I don't think that we're ever going to get away from that in the same, like maybe if we have quantum computers that can like perfectly simulate, biological systems, like fine. But I feel like you're always be doing experiments. And so you're always going to be thinking, how am I like making sure, so that will always be your latency. How do I make sure that next experiment is the best possible one? And so you're gonna have to do some inference time work for that. And I don't know that that's always going to be in an RL loop, right?
No, I totally agree with you.
Like you could, like there are some people who do think that. I'm just curious.
No, I totally agree with you. And I think there's also, just to get back into the RL loop, there's different types of RL loops you could run. There's an RL loop where you teach the thing to use tools. There's the RL loop we tried out in RBio, which is make the thing reason as a biologist directly without using tools at all. RBio is tool-free, doesn't use tools at all, not even during test time. So it's static in that sense, but it has distill the knowledge from the models. And there's combinations thereof in the future that
it does
during
call a
training
model at some point.
it calls the models to learn,
Okay.
but during testing it never calls models anymore. But you could
Sure.
train, and many people have done that,
Sure.
an RL agent to use tools better. And you could do all of the above. And I totally agree with you that what you ultimately want to do is you want to use test time compute to search hypotheses so you can pick the right one to go do in the lab and verify, without a doubt. And you want to do that in a way that is, so to speak, base optimal. So you go choose the right thing to do that is the most likely thing and that you learn the most from in case you were wrong, because you might be wrong, you might be miscalibrated. By definition, you should be wrong. And your goal should be to then, falsify your hypothesis as well as possible with the experiment until you can't falsify no more. And if you falsify enough hypotheses in the lab in an optimal way, you might be able to actually make discovery progress. So I don't disagree with you at all. And the question is, how do we engineer this with the lot of different tools and techniques we have?
And have you seen this lab in the loop working for any particular task efficiently? Because this is something what we have been talking about as a field for many years now. But I still feel when I talk with anybody that it's an open problem, that how you make this efficient. Have you seen this?
I think it's very early days. Lab in the loop is kind of a very big topic where people think of robots automating everything. For me, lab in the loop fundamentally is the idea that we can automatically transition between hypotheses. And I'm personally still totally fine if humans ran these hypotheses in the lab. So I want to distinguish between, is the lab automated or is the brain that generates what we're supposed to be doing automated?
In this case, I mean the latter. So it's not, it doesn't matter if it's an automated lab or not, but you have a set of hypotheses, you measure them, put that back, learn, and do it again and again.
I haven't seen that rigorously been tried yet because there's so much new stuff in there, right? People have done these wonderful discovery loops, like the people at Prescient Design, that clearly have led to antibodies and so on. That's wonderful work. I'm very excited that some of this has trickled to the outside world and we've seen it. But I think this is still very new, too new to judge. I'm excited about what people will do.
So I definitely, so in the world of medicinal chemistry, these like DMTA, design, make, test, analyze is actually like fairly, like that definitely is in production today at scale for very, very simple properties. So if you have like a single scalar readout assay, like think like, I don't know, like viscosity or like a physical property of a substance, and you have like a relatively well understood like set of things that you can go and optimize over, like there's certainly fully autonomous loops for that. However, they're kind of all just Bayes opt. Like if you have that well of a defined system, you can just do classical Bayesian optimization over the hyperparameters and get there. I think that the like interesting part about agentic discovery is that, I don't know, in my mind, the thing that makes agents really interesting, it's sort of like the, it's like universal function approximator theorem for deep nets. Like I like things where you sort of like have the outer bound. Like if a deep neural network is sufficiently large scale, can approximate any function, like that makes it an interesting substrate to study. In the same way, like agents kind of have an unbounded search space over the world of bits, at least. And so like, ideally in a software engineering like optimization loop, like you could optimize over literally everything, the agent itself, all the tools it has access to, like, anything that you can actually put within this sort of touch of it, it can go and work over. In the world of atoms, it's a little bit more complicated because like someone needs to go and like buy the chemicals. And it turns out that like even just the problem of like what chemicals are available to purchase from a vendor is not there's no like API necessarily, or there's like you have to have a human interaction in the loop there. And so I just think like, so that's like one sort of area of friction. The other one is that I just don't see a lot of people, like there's some hesitancy still, like lack of trust. Like, I was talking a little bit at the beginning, like you have to have like doing the single cell analysis, you've got to like talk to the biologists, like make sure that they trust what you're doing. There's still so much of this like cross-disciplinary work. And I don't think you can like truly get that like feeling of like, oh, this is like fundamentally changing how the world is going to function if you don't use it yourself. And so there's still these like barriers where I see groups that I would think, oh, like, of course they're doing a fully lab in the loop process for designing like molecule X. And then you talk to them, and they're like, yeah, but there's still like the holdout who like wants to review the list of experiments and like say, oh, this one is the one that we should or shouldn't do. And you're like, but why? Like you have a model, like train the model, like just take the full like, you know, AI pilled approach and just like play it out and see how it goes. Otherwise, like you're never going to be able to understand the process. You can't optimize the process.
But but I think, Daniel, to your last point here, this is a transition that we will experience, I believe, soon. In the past, when people had
Yeah,
models for these things, the models were focused on the thing that they built the model of, which are few things. As we are now in this agentic universe where these things are extremely powerful and nearly unbounded, as you said, they understand a lot of different things and so on. It becomes way, way, way easier to actually start having a search space in them and a conceptual space where they represent a lot of what's actually going on in the experiment. So it's a broader capture
yeah,
of the data of what's happening. And I believe in that world, it'll become easier to represent the protocol, the variation, the many different things, and actually start iterating on these things usefully. In the past, this is about fundamentally, if I can just call it what it is, in machine learning speak, the models in the past, the hypothesis space were underspecified. So fundamentally, your model is
yeah,
misspecified if you just can't identify a set of parameters that will characterize the solution that you care about, right? You can't get there. So these poorer models in the past were misspecified. This is why there needed to be this manual labor, the manual layer where you really go in and you say, not this, not this, not this. And this creates distrust. We are now in a space where maybe we're still misspecified, but surely a lot less. If anything, the problem now is the hypothesis space is so gigantic that search is the problem because you need to have enough data to identify these things, which we're learning to constrain these things in various ways. But I think as people realize that anything they can express in language is expressible in these things, we will find out that we will start using them more and more to commit to these lab in the loop type things. But I personally haven't seen that fully happen yet because it's new. Everything we're experiencing is very, very hot off the press, so to speak.
Yeah. Let's double click a bit on this skepticism, because even in the Q&A there are multiple, I guess, computational biology background people asking about batch effects and how do you see the data and all of that, and this being one of the limitations of utilizing agents to their full extent. And my personal experience with the team was when Biomni came out a couple months ago. The team tested it, and this was the first time that they were actually surprised at how good it is. But they also told me that they were prompting it based on our experience on how to set up the workflow. So it did what we usually do, and that already contains the learnings of how to do computational biology. So the question to you, and I guess first to Daniel and then Theo jump in, is where is the right place of the human in the loop? What do you expect from the human to know? What is that expertise which is still needed to run this efficiently? And then you can just trust it, as you said, Daniel, and you don't need to overthink it. Just test what comes out in the end.
yeah. I mean, like, there's no one answer for that, right?
Sure.
Like, it's always every system, every process is going to be different. And I feel like it's also like one of the messy parts about all of this is that at some point, like, you have to spend money. And so to get money, someone has to give it to you generally. And people tend not to just give money without strings attached. And so like, and then to do things, you often still need other people. And so like some organizations, some funders just are like going to be more or less predisposed to like more or less hands-off work. Um, like some groups really like to build everything in house. Some people like to outsource everything. Like I think to some extent, like that, I think groups that are more amenable to using CROs are probably going to be more amenable to outsourcing some of the intelligence to models, which I think is good in some cases.
Mhm.
Um, yeah, I don't know. Like there was a question I'll just maybe answer directly, which is like, how do you define a reward signal for a scientific
Mhm.
agent when the value of a negative or unexpected result is so context dependent? And the answer is just it depends. Like I'm sorry that there's no like clear, straightforward answer here. This is why like I still think it is a good idea for people to go to university and go to grad school. I think that learning how to like work with people and reason through uncertainty and complex problems, like I'm like debugging like an open clock cron job, like context snafu that came up this weekend. I'm like, how is this a problem that I still need to intervene in, where like a cron job is failing because it's not picking up the right context when it's like somewhere in the agent system? Like still I need to intervene in systems like this. And so like we're just not close to the point where, oh, there's a clear like here's your handoff point, or like here's exactly like the one rule for crafting a reward signal, because if there was, then like you shouldn't have a job. If I can tell you in one sentence like where the line is, like that's not a good job to go have.
I will try to answer this in a way that I don't talk about jobs. The beginning of the question was, remind me, what did you start answering, Daniel, and then you got to that question that was about the reward?
It was about like where's the point of the human in the loop?
Right. Yes, apologies. So I think of this a little bit like in education. You want to give them roots and you want to give the agents wings, and you want to give them both. And you said the smartest thing that anyone has said in this panel, which is you still need to make money and you need to do that in a bounded amount of time, which means the role of the human is to fundamentally provide guardrails and sanity for these systems early on, because they have an unbounded search space and could do random things and consume a lot of resources and waste a lot of time and get nothing done. And that is the default state one would expect. And so in the beginning, you help them think through strategy by correcting them a lot, by doing a lot of this. But as we've seen also with coding agents and skill and so on, these become memories, this becomes data itself, and agents learn from that. And ultimately, the role of humans will be to correct these objects early on, and later on to teach them to think, teach them strategies. And I think as we teach them strategies, there's a notion in AI for that. Eventually, what you're doing is you're aligning them. Eventually, what we will do is we will align our scientific agents to behave the ways we want them to behave, because the real reinforcement signal they get from the real world, which is how often do you do discoveries, is very sparse. It's a very delayed reward, sparse reward signal. You can't rely on that in some automated way to make them do what you want. You really need to provide the intermediate signals. And as humans, these are very hard for us as well. How do we learn as humans to do science and to invent things? An entire education system that teaches us to reach partial goals of high value, learn truth, establish truth, build generalization, really bridges from one thing to the next. If this is a fact, then this. It's logic, it's reason. These things are difficult to do. And they're not going to come overnight, that agents will just become scientists. And I think humans, as Daniel said, are still very important. And I think of this a bit as programming. This is just a higher level layer of programming, and we're learning to program the scientific process a little bit, and the agents take on a layer of automation, but they're not fully autonomous scientists yet, because the amount of reward signals they will get are still very poor.
And I see this came up also in Daniel's presentation on why do we do things, or what is the question you ask is primarily a human thing, and then how you address it can be driven by the agents and these different models. And one like key aspect which came up in the questions as well, how can we utilize these tools more? One question, which is a human question, is the build or buy, which you had, Daniel, also mentioned. So if you want to give like practical advice to the audience, and you are, let's say, a big pharma, or you have some money to spend on these technologies, then what would you build internally and what would you buy? And again, I know there is no one answer, but what would be your take if you would be in the shoes of, I don't know, AZs, the Mercs of this world?
Yeah, no, I've got a good answers for both. The first thing, I guess it makes kind of similar. So you just need to use these things more. I think that like simultaneously, agents are the most transformative technology that I think we've seen in the past, 20 years. And also they don't really change much. And I think like understanding that tension took me a very long time to get towards. Like, yes, everyone now can have an army of interns that they can spin up if they can pay for the tokens. But like that doesn't mean that like everything about the way that we work or ascribe value to things or anything changes. It just means that people get more leverage, like a lot more leverage, but not fundamentally different. So I just use them. Like go to ChatGPT for everything that you wanted to go and do. If you don't know what to do, ask ChatGPT, hey, what is something I could try to do to like use AI for something more in my life? I don't know. There's like so many ways to do this. And then I think for the large pharma companies, like honestly, the biggest advice that I give is like very similar, but it's more around the fact that there's a bunch of emerging products in the space. So the product surface for agentic platforms is incredibly immature. There's like basically chatbots that work kind of okay. There are AI coding harnesses that also work fairly well. But the like things on top of that are just like very, very young. And so I really think that the most important thing is just to try them all. Like I use ChatGPT and Claude. I use multiple voice AI tools. I use multiple different kinds of Claude things, like just to try them because like otherwise you won't know how they work and you need to understand sort of the surface of them. And it's not really that expensive. Like, yeah, maybe more than your enterprise SaaS stuff, but like it's not like it's not like a CapEx purchase. You've got to go spend like $50 million to go and get your new, like AI tooling for your company.
Do you any comments on the build or buy?
I really, really like the idea of trying these things a lot when you're in, and there's a very fundamental reason for that, right? Right now, the world is changing, at least the digital world is changing faster than ever in my life. And every couple of weeks, there's something that feels very different. And from a fundamental, system identification point of view, when your system that you're exposed to changes very fast, what you want to maximize is the amount of times you interface with it to learn. So your own world model of that system tracks. And as your own model of these things and how they behave has bias, it clearly has bias. No matter what model you're building, all models are wrong and your models are biased to the data you've seen. If your external world changes quickly, your bias will take over if you don't keep interacting to update your model. So I think you need to, in this day and time, it's really, really valuable to explore and to use things a lot, get a lot of experience, try rigorously, get your hands dirty in order to understand what's going on and how things are changing. And eventually, I think the richness of the tooling out there and what's being created will yield something useful to people. I would just say this more so than ever is not the time to say, I have a thing I like and I'll stick to my guns. I think this is a time where we can really dramatically rethink how we do things and make them better. But you can't chase hypes. You can't always just do the next thing that people find cool, but you can try a lot of different things to find out for yourself, your purpose, your work. What is the right tool? What is the right thing? And even if you say, I don't trust these things, they didn't work for me 2 months ago, it may be very different now. It may be very, very different now. So I just think this is the moment we're in.
Yeah, the rapid changing pace is insane. Like I remember last summer trying to navigate iClear using LLM to score papers, and it was like I wasted like $500 in credits. It didn't work. This year, it was flawless. Like I will never go to a conference without having an AI read every conference abstract and pick which posters to go to. It'd just be a waste of time.
Yeah, let's close on that. I think the most practical advice we can give to everyone is to try these things. Many of these are at your fingertips, and even like the open models you are publishing, Theo or Daniel, are usable. So it's not just the LLMs, but also the different virtual cell models are available. So you can, in pharma, but even biotech or even like individual researchers, you can try many of these and build an understanding, and then it will be much easier to implement them when the new technologies are coming. Thank you both. This was a very I don't even know what's the right word. We tackled a lot of ground, and it was very illuminating for me, at least, to have this discussion. I will just close with a QR code so you can, the audience can give us feedback and also subscribe for the next meetup in the series. Thank you, Theo. Thank you, Daniel. This was great. And hopefully see you soon. Thank you, everyone.
Thank you for the invitation.
Bye-bye.